In the last video we spoke about differential PCR, and by that I mean just the amplification of certain alleles according to which primers you put in. Let's look at a further application of that, and that application is gel electrophoresis, i.e. the separation of mixed samples of DNA. Let's say we have someone who we know is heterozygous. Okay, so this person is heterozygous and they have two different alleles on their two chromosome 1. So, for example, on one strand here they've got allele 2, and one strand here they've got allele 1. We throw in those two different primers, so those primers will happily amplify. So primer 2 will happily amplify the orange allele here, and primer 1 will happily amplify the green allele as well, and we end up leaving it for a while, and you end up with a big mixture. There'll be loads and loads of strands of the two different alleles and those two different bits of DNA. So you end up with loads of DNA, and that's fine and that's great. The problem is they're mixed together, and you will want to separate them. You don't. Why you need to be able to do that doesn't sort of kick in until a second, but you will need to. And the way you do that is with gel electrophoresis, and what it is this big gel. It's like a slab of gel. It's a very solid gel, more like a jelly, made of agarose. And what you do is you put a current through it. So you have a negative current, a negative end and a positive end, and you load up your mixtures into these little wells over here, and then you allow current to pass through the gel, and that will happily drag the DNA molecules that you have through the gel. And because DNA with all its phosphates is negative, it tends to go further towards the positive end. So you leave it, make sure the positive is on this side, and think that the DNA will naturally move towards the positive. Thing is, everyone's like, how do you get a separation from that? And the short and easier answer is the shorter pieces of DNA. So in this example I've purposefully made allele 2 shorter. If you notice, the shorter bits of DNA find it easier to get through the sort of thick, slightly strange agarose matrix, and they will therefore pass further. So this would, if you have a mixture, this would be the shortest, and this would be the longest piece. So for the example I've given you, the orange was shorter, the green was longer, and therefore if you'd left it and you'd loaded it into just this well here, you would get something that looks like so. In terms of other things you need to know about this, is this doesn't happen in isolation. You need to bathe it in a fluid, and the fluid that you bathe it in is a buffer. Okay, so this keeps pH very stable. And that means that basically if you have fluctuations in pH with DNA, it can interrupt hydrogen bonding, and there's some evidence that it can also sort of break the hydrolyzed phosphodiester bonds as well as sort of doing the whole process. DNA is clear, you can't easily see it. In all of these staining, I'm color coding it to make life seem easier for you, so you can see which is the long and the short allele that would be split up. In reality, you need to stain them or dye them, and you use something called ethidium bromide to do that, which is hugely toxic. Equally, you can use sort of UV for certain samples, or you can even use radioactive tags, so radioactive labels, and then that way you can detect the radioactivity that comes off them. So that those radioactive labels stick to DNA. You can then detect the radioactivity just sort of like photography paper like you saw in GCSE physics. And that's effectively it. When would you use this? So we've talked about sort of like having these mixtures of certain things. It doesn't really make sense. Let's say you have another individual who you've just run that gel for that person. You've got the green and the orange, so you've got a nice big sample of that you could do again. Let's say you've got their sibling and you want to know whether they are heterozygous, homozygous for one of the genes or homozygous for one of the others. You can do the exact same protocol where you do PCR, with all of those primers within it, and then you can run a gel electrophoresis on what comes out. So say for example you end up just getting another splodge that is very similar, that should be almost identical to it, how far along it goes, then you know that they don't actually have this allele, so they must be homozygous for whatever allele this is, and you can start to use it to actually get out some ideas of people's genotypes. That won't make full sense until you've done patterns of inheritance, but there are certain questions in the past paper pack or that we've done in class that test this sort of knowledge. The other things that you quickly need to know about it is sometimes you will end up with very unknown samples. You don't fully understand the gene that you're looking at. You just know that you can amplify it, cut it up, and then want to run it. So if you were looking for some clues about what this gene might be or how long the gene might be that you're looking at, if you're trying to sort of identify a new gene, you can run it with some primers and get some idea of how long it is. And what you do in order to give yourself some idea of length of the samples that you've attained is that you actually run it against a known sample, for example. So here what's happened is they've loaded up the wells and they're running this way. So this would be the positive end and this would be the negative end. It would run towards the positive. The longer ones stay closer towards the top and the bottom ones travel further, but you have what's known as like a marker sample, and this is something you can buy off the internet, which is a mixture of DNA fragments that are 1,000 base pairs, 900 base pairs, 800 base pairs, 700, 600 base pairs, so on and so forth. And you can start to see like, this unknown thing that I don't know, or these genes that I'm amplifying has got something that's like 1,000 base pairs long, there's one that's 900, there's a 700, like what's that, 450-ish, and 200 as well. So it just provides some clues, and that's using a marker sample to guide your thinking. The last thing you need to know is if you notice you don't really go above 1,000 very much. I don't know the exact number, but you can't use gel electrophoresis for particularly long. You can't use gel electrophoresis for particularly difficult or long or complex DNA fragments. You sort of get limited at 1,000. The reason behind this is this graph. So the size of this is how far DNA fragments will travel in gel electrophoresis depending on their size. As you get past a certain point, you get to sort of a plateau. It's not zero, it's still moving, but they move at a chain that is 5,000 base pairs long as opposed to 6,000 base pairs long will not move a hell of a lot further or as quickly as a 6,000, so that they're very, very similar. So if you were to run them next to each other, you end up with very, very little resolution between the two of them. If anything, I've given this too much credit. They would be basically on top of each other, because as something that's 5,000 or 6,000 moves at the same speed, if you leave them for an hour, they're just going to end up basically in the same place. You could say, could I run it for longer over a massive, massive gel, and that will sort of make the differences more stout and clear? You could do that, that is true, but realistically the exam board just wants you to know that you're sort of limited by how big DNA sizes you can do. Okay.